The next topic for exam 3 is immunology, or the immune system. And we'll talk about both the innate and adaptive immune responses. So if you're wondering right now, why are we talking about the immune system? I thought this was microbiology. Well, they're very closely related, especially for this class where we're interested in medically important microbes and how we as humans interact with microbes. And if you got to this lecture, you probably already saw the infection and infectious disease lecture, learned about what pathogens are. So now we'll see what our body does whenever a pathogen is introduced into our system. And we'll zoom in into that microscopic level, because microbes are microscopic, and we'll see how, on a microscopic level, what are our cells doing in our body to combat those pathogens. So over this lecture, we're going to talk about the two branches of the immune system. We're going to talk about the innate or non-specific immune response and the adaptive or specific immune response. And we'll start off by talking about the innate immune system. I'll just do a little overview of these two systems so you know what to expect throughout the lecture, and for a lot of you this may just be a review. So the innate immune system is our rapid response. It occurs within minutes or hours of something foreign being introduced to our body. It is non-specific, so anything that seems foreign to our body, our innate immune system is like, get it out of here. It is response fixed or non-adaptive. So it has the same response every single time something foreign is introduced to it. And there's no memory involved with the innate immune system. The adaptive immune system, on the other hand, is a delayed response, so it can take days or longer for the adaptive immune system to mount a response toward a specific antigen. So it's highly specific against very specific foreign molecules. It is response adaptive, so it changes over time. How it responds to a certain pathogen or foreign molecule will change as it learns what that molecule looks like. And it has immunological memory, so it remembers what a certain pathogen or foreign molecule looks like, so it can mount a specific response in the future. So our immune system is broken down to the branches of the innate and the adaptive immune systems, but we can also think of it in terms of lines of defense. So there's the first, second, and third line of defense of our immune system. And this will come in handy as we go through, just so you can keep track of what we're talking about, which immune system we're talking about, and all that. So the first and second lines of defense are part of our innate immune system. We're going to see in the next few slides that the first line of defense is talking about our physical barriers, and the second line of defense is talking about the chemical barriers and some inflammatory responses with certain cell types. But again, the innate immune system is all non-specific. And then the adaptive immune system is our third line of defense. This is where it gets to immune system specificity and creating an immune memory for future pathogens. So the innate immune system, this is where we're starting. And as I've already mentioned, the innate immune system is non-specific. This is the response that occurs immediately or within hours of exposure to almost any type of microbe that your body sees as foreign. We'll talk about different types of non-specific host defenses that you have, like the physical and chemical barriers, fever and inflammation, our normal flora, and genetic barriers. So these are all examples of non-specific host defenses that are part of the innate immune system. Now we're within the innate immune system, and the innate immune system includes both the first and second lines of defense, and we're starting with the first line of defense. And our first example is this physical barrier of our skin. And if you think about it, wave your arms around, think of all the stuff you've touched today. Our skin is absolutely our first line of defense against microbes that we're coming into contact with every day. And our skin plays a lot of important roles. We'll talk about both physical and chemical roles that it plays in preventing infection from pathogens it's coming into contact with. Another first line of defense or non-specific immune response defense are our mucous membranes, and we'll talk about that after we get through skin. Our skin is built to protect us. It has physical components that defend us from pathogens just being able to enter right into our body. The skin has two major layers, the epidermis, which is the outer layer, and then below that, the dermis. The epidermis on the surface has these layers of very tightly packed cells, so that prevents pathogens from just entering into our body. On the surface of our skin is also a layer of dead skin cells that are constantly being removed throughout the day. And so pathogens or microbes that are attached to those dead skin cells just leave with the dead skin cells. That helps reduce the microbial load. And then also in this epidermal layer, you have dendritic cells that help phagocytize or eat and break down pathogens on the surface. Below that, the dermis has these protein fibers called collagen, and this helps give the skin a lot of strength and pliability. So this means that whenever you make a fist or you bend your elbow, your skin has some stretchiness to it, it has some strength and durability, so every time you're bending your arms, you aren't breaking your skin open and giving a clear path for pathogens to enter into your body. Our skin also has really important chemical components that defend us from pathogens. So it has both physical and chemical components for protecting us from infection. First off, our skin has sweat glands. These sweat glands help prevent colonization by pathogens two ways. So one, sweat has a high salt content, and this inhibits the growth of a lot of pathogens by desiccating or drawing out water from the cells because of that high salt concentration. And then sweat also contains a special enzyme called lysozyme, and this will come up again and again because this is a common enzyme our body uses in a lot of our secretions in order to help prevent the overgrowth of pathogens in our body. But lysozyme is in sweat, and it destroys cell walls of bacteria and reduces the microbial load. Also, our skin has oil glands or sebaceous glands that secrete sebum onto the surface. And this helps with a couple things. First off, it helps with that skin pliability, to moisturize your skin, to have it less likely break and tear whenever you are moving your joints. And also, sebum lowers the pH, and that fluctuation of that slightly lower pH does inhibit many bacteria from being able to survive on surfaces of your skin that are more oily or sebum heavy. Now we're still talking about the innate immune system, and we're still talking about the first line of defense, all right? So we talked about skin, and now another first line of defense is our mucous membranes. Our mucous membranes are going to line all of our body cavities that are open to the outside environment. So think about your digestive tract, and your nose, your respiratory tract, they're all lined with mucous membranes. And as we saw with the skin, we have that outer layer of dead skin cells on our skin, but the mucous membranes are consisting of living cells. Even though those living cells are continually being removed and shedding away to get rid of attached microorganisms. Very similar to the surface of the skin. And also similar to the surface of the skin, the cells that make up our mucous membranes are tightly packed to prevent pathogens from just being able to pass that skin barrier and enter our body. So like our skin, our mucous membranes also have physical and chemical defenses to protect us from pathogens. Our eyes produce tears, which also have that enzyme lysozyme in them. We are blinking to remove debris from the surface of our eyes, and eyelashes to whisk debris away before it gets into our eyes. The genitourinary tract, especially in women, usually has a lower pH to prevent the overgrowth of microbes. And then also the flushing of urine through the urethra to remove the microbial load, the production of mucus from those mucous membranes. Our respiratory tract, and we'll talk about this more on the next slide when we talk about the mucociliary escalator, but our respiratory tract produces a lot of mucus because think about it, you are constantly breathing in, you have to breathe in, and you're interacting with the air in your environment. That air may or may not contain different types of debris or pathogenic microbes. And so having that mucus there and the mucociliary escalator traps those potential pathogens and removes them before they can enter further into the respiratory tract. And then finally, the GI tract or digestive tract. So this also has that special enzyme lysozyme. This is going to be present in our saliva and different secretions within the GI tract. And then also your stomach. Your stomach is a very acidic environment that is going to prevent microbes from surviving once it gets to the stomach. Now let's talk a little bit more about the mucociliary escalator and the role it's playing in preventing pathogens from reaching deeper into your respiratory tract to your lungs. So you'll see this diagram on the left. It says the trachea. The trachea is this tube that is protected with cartilage, and that is your windpipes. That's what is allowing air to get down into your lungs. And lining your respiratory tract, you have mucous membranes. And these mucous membranes have these special cells called goblet cells that secrete mucus onto the surface of the cells. And that mucus is great because it's nice and sticky, and so it traps a bunch of things that you're breathing in. And when you think about it, you're interacting with the air around you all the time because you got to breathe. So you're breathing in stuff, and if you're breathing in certain particulate or pathogens, then it'll get caught in this mucus. And then the cells that line the mucous membrane have these projections on them called cilia. These cells have cilia sticking out from them. And these cilia move or beat to push the mucus up, up, up. So mucus is moving up, 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 like an escalator. It's propelling it upwards. So that mucus is getting filled with anything that's getting caught in it as you're breathing, and it's moving up away from your lungs, and then eventually that mucus will leave by you coughing up phlegm or blowing your nose, but most often you're just going to swallow it. It'll go down your esophagus to your stomach, and then disintegrate in with your stomach acid. So the mucociliary escalator is a very important part of the respiratory tract for limiting the number of microbes that are getting down to your lungs. Because really your lungs have very, very, very few microbes. Really, it's one of those areas that really shouldn't have any normal microbiota. So the mucociliary escalator does a really good job of protecting your lungs from infection. And then finally, some other first line of defense barriers and kind of moving into second line of defense. We have our normal flora, which we have talked about already. Your normal flora or normal biota, normal microbiota. That's all your wonderful bacteria and other microbes that live on your body. And they protect you by taking up space and nutrients so pathogens can't reside on or in you. But also they do produce certain enzymes that kill bad bacteria like colicins and beta-lysins as well. And then certain people have genetic traits that protect them from certain infections as well. Interestingly, people with the sickle cell trait do not get malaria. They are not infected by Plasmodium falciparum because that parasite requires access to a red blood cell in order to replicate as part of its life cycle. And the sickle cell trait changes the shape of the blood cell, so the Plasmodium falciparum cannot use it to replicate. And then finally, acute inflammation. This is taking us into the second line of defense because inflammation kind of bridges the gap between first line of defense and second line of defense. And we'll see that whenever we enter into second line of defense stuff. But inflammation is really important because it is non-specific. Anything that your body sees as foreign will elicit the inflammatory response to alert your body something's wrong and then bring resources to the area. Now on to second line of defense, and we're still in the non-specific immune system, so the innate immune system. And the second line of defense is the result of pathogens succeeding to penetrate the skin or mucous membranes. So damage has happened. There has been a breach in the barrier. Now we've entered second line of defense, and now we've really entered the inflammatory response as that primary second line of defense. And the second line of defense involves a lot of things. So it's going to involve the cells involved in the inflammatory response and the certain types of chemicals and antimicrobials that those cells produce. And then those different processes and cascades involved in getting all of those cells to the area where there is damaged tissue. Now let's talk a little bit more about the inflammatory response. And you should know the cardinal signs of inflammation. So heat, redness, swelling, pain. And we've all experienced inflammation before, right? And it's a really important part of that second line of defense of the innate immune system because it's your body's immediate response to something's wrong, something may be in the body that should not be in the body. What are we going to do about it? So here are the steps of inflammation. We have some damage to the skin that's happened, so we know we're in the second line of defense because this barrier has been breached. And then the damage to those cells is going to elicit an immediate response to release cytokines. Cytokines are chemicals that cells release so they can communicate with each other. And one of the chemicals that they're releasing is histamine. And histamine elicits this inflammatory response that's going to cause capillaries to dilate. So they're going to widen and allow more blood flow to the area. And that they're also going to become leaky. And that's going to cause that heat and swelling and redness in the area. It's also going to recruit a bunch of white blood cells to the area and clotting factors to help close up the wound. One of the white blood cells that they are calling to the area are different types of phagocytic cells. Phagocytic cells are white blood cells that are going to really consume the bacteria or whatever pathogen or bad thing has entered your body. They're going to consume it and break it down. So these are the steps involved in the inflammatory response. And you can see how it is non-specific. So anything that enters through that barrier, through that first line of defense barrier, is going to receive this same treatment of the inflammatory response. This is just a closer look at what a phagocyte is doing to those invading microbes. And we're going to see this when we talk about different types of white blood cells, but the two phagocytic white blood cells I'll ask you about are neutrophils and macrophages. So in both cases, here's the white blood cell, the phagocyte, and it's going to recognize something foreign in the environment. And so it's going to take part of its membrane called pseudopodia, and it's going to extend that membrane and the pseudopodia to encapsulate the invading microorganism, forming a phagosome. So it's trapped inside the cell. That phagosome is then going to connect with something called a lysosome. And what enzyme does a lysosome contain? It contains lysozyme. And we know that lysozyme, we talked about it's in our tears and our saliva, and that's important for breaking down bacteria. So that's the same enzyme that phagocytes are using to break down bacteria as well. These two will bind together and to allowing the lysozyme to break down the bacteria within the phagocyte. And then over time that will be broken down, and it can take hours for it to break down inside the cell. But at that point, the white blood cell can then release this waste out into the environment. Now let's take a little detour. We're going to talk about the blood. We're going to talk about different parts of the blood, and then we're going to talk about different types of white blood cells within the blood. And this may be a review for many of you who have taken anatomy and physiology before. But there are different formed elements within the blood. So we have erythrocytes, or red blood cells. Those are the cells in our blood that are responsible for carrying oxygen to tissues and carrying away carbon dioxide. There are platelets in the blood, and this is a clotting factor, so it's important for the inflammatory response and closing up wounds. And then we have leukocytes, and leukocytes are white blood cells. So that's interchangeable. Leukocytes, white blood cells. And this is really important for our immune response. So leukocytes are involved in defending the body against invaders. And there are two types of leukocytes, granulocytes and agranulocytes, and you'll see the difference between those on the next slide. But just know the general formed elements. I'm not going to ask you too much about erythrocytes or platelets, but definitely we'll go into more detail on these leukocytes. So leukocytes, or white blood cells, WBC, are divided into these two major groups. We have agranular and granular. And I'm not going to ask you too much about these two groups. I just want you to be aware that it's one way of grouping them. Really, they are given this definition based on what they look like under the microscope. So granular leukocytes look granular under the microscope. They look like they may be filled with sand, whereas agranular do not appear that way under the microscope. And whenever we do get to look at a white blood cell or blood smears in the lab, you may be able to differentiate the two based on the agranular or granular appearance. Now we'll talk about the different types of white blood cells that belong to those two groups. First off, we have granulocytes. And the three types of granulocytes are neutrophils, basophils, and eosinophils. And each one has a special job. Neutrophils are definitely your most abundant white blood cell in your body right now, making up to 60% of the white blood cells that you have. And if you've ever gotten hurt or had a wound that you take off the bandage and you see there's all this pus around the wound, those are actually white blood cells or neutrophils that have died. So that's all of those white blood cells were sent to the area through the inflammatory response, and then they accumulated and did their job and then died. So neutrophils are one of those phagocytic cells as well. So they're one of those first responders to the inflammatory response. And then we have basophils, and this is the least common white blood cell. Basophils are important for releasing histamine and other chemical mediators that are part of that inflammatory response. Like I mentioned, cytokines are how these cells communicate with each other. So basophils are important for the release of histamine. And then eosinophils play an important role in fighting viral infections and types of parasitic infections, like infections from helminths or parasitic worms. And then also eosinophils can phagocytize pathogens as well. But I'm really only going to ask you about neutrophils and macrophages as far as phagocytic white blood cells. Now we're on to agranulocytes. The two types of agranulocytes are going to include lymphocytes and monocytes. And lymphocytes are really involved in specific immunity or adaptive immunity. Lymphocytes are going to include your B cells, T cells, and natural killer cells. And we'll get to those later on when we talk about specific immunity. But just remember that we're still talking about leukocytes. They're still a type of white blood cell. And then monocytes right here, as they mature, they become macrophages. And macrophages are important phagocytic cells, also for that second line of defense and being recruited to an area of inflammation. So let's put everything together that we just talked about over those last few slides. So remember, a leukocyte is a white blood cell. Those are interchangeable with each other. Leukocytes are broken down into two groups called granulocytes and agranulocytes, and it's just based on what they look like under the microscope, whether it looks like they contain granules or not. The types of granulocytes include basophils, eosinophils, and neutrophils, with neutrophils being the most abundant white blood cell you have in your body. And then agranulocytes include lymphocytes and monocytes. Monocytes mature into macrophages, which are an important phagocytic cell, along with neutrophils. And then lymphocytes are really important for the specific or adaptive immune response and include T cells, B cells, and natural killer cells. So remember that all of these right here that I'm going around with my pointer right now are types of white blood cells. They're all types of leukocytes. On the next slide, we're going to talk a little bit more about natural killer cells because really they kind of bridge the gap between non-specific and specific immune responses. Sometimes they kind of just like hang out in this gray area in between, but really they're considered part of the non-specific immune response or your innate immune system. So that's why we're going to talk about them now in the second line of defense discussion before we get to the specific immune system. Natural killer cells. Natural killer cells are lymphocytes. They are a type of white blood cell. They're also called NK cells. Natural killer cells kill other cells by secreting toxins called perforins. These perforins, just like it sounds, perforate the membranes of other cells, breaking them down and killing the cell. Natural killer cells are really going to be effective against virally infected cells or tumor cells. So they're working on mostly your somatic cells, whether they're infected with a virus or they are altered in some way due to a tumor. They're going to recognize which cells to kill and release toxins for based on the MHC1. This stands for Major Histocompatibility Complex. And we're going to talk, this is going to come up again, so don't worry about it too much right now. But really what natural killer cells are looking for is this this tag or this indication on the surface of a cell that says, yes, this is my cell, or no, this isn't my cell. Cells that are infected with a virus or are tumor cells have low MHC1 and they don't really look like your body cells, and so natural killer cells can come along, identify that, and then get rid of infected cells. Interferon is another important part of our second line of defense. So interferon is another type of cytokine, or a way that cells communicate with each other. In this case, interferon is a cytokine that's produced by cells that have been infected by a virus. So you see in this image, here's the cell, here's the virus. It has bound specifically to its host cell type, and it has infected the cell. And now this cell is like, oh, geez, something's wrong. I better tell my friends that are around me. It's going to produce interferon, or the cytokine that it uses to communicate with other cells, release that into the environment. That interferon's going to bind to the other cells in the environment and tell them, hey, look out, there's a virus. Don't get infected like I did. And so it's going to tell these cells to activate certain things to prevent this virus from being able to bind and enter inside the cell, making them resistant to a viral attack. Now, the last thing that we'll talk about for the second line of defense, or your innate immune system, is complement proteins. Complement proteins are 25 to 30 proteins that complement the work of your immune system. Really, they're complementing the work of antibodies and your adaptive immune system. They're constantly just circulating in the blood in an inactive form until a cascade is activated by this primary protein being attached or activated, and then causing this cascade. What I'm going to ask you specifically about the complement cascade is not any of this. You don't have to know all of this. This looks like a complicated image, but it's not that bad. I just want you to see that it is complicated, that there are a lot of proteins involved with this. And the complement cascade ultimately can form something called a MAC. This is a membrane attack complex, and I think it's very fascinating. So a membrane attack complex is the result of this complement cascade happening after it's being activated by there's the presence of some type of pathogen. Something shouldn't be in your immune system. This cascade is activated and forms this MAC, which is really a ring of these complement proteins that form to sit down on a pathogen and literally bore a hole through its membrane. So it's a membrane attack complex. And of course, once its membrane has been compromised, it kills the cell. So it's a really fascinating thing that complement proteins can do. But they have other jobs too. Like they can recruit phagocytic cells to an area. They can also help activate the inflammatory response. So they really are an important part of complementing that inflammatory and immune responses. This is just a summary of some of those non-specific components that we just went through. So non-specific is including both the first and second lines of defense. And just remember that all of these that we just talked about, we were talking about the innate or non-specific immune system. Just a practice question here if you want to pause. Macrophages arise from which of the following? The answer here is monocytes. So monocytes develop or mature into macrophages. And macrophages are important phagocytic cells, meaning that they consume or eat bacterial cells and then break them down. The other phagocytic cell that we are interested in is neutrophils. So now we're going to move on to the specific immune system, or the adaptive immune system. So now we've talked about the innate or non-specific immune system. We've talked about the first line of defense, which is just our barriers, like our skin and mucous membranes. We talked about the second line of defense, what happens when that barrier has been breached. And now we're going to talk about the third line of defense and the adaptive immune system and immune memory and antibodies and antigens and all that fun stuff. Our third line of defense now includes our specific or acquired or adaptive immunity. Those are all interchangeable. And our specific, acquired, or adaptive immunity is going to be our body's ability to recognize and defend itself against specific invaders. So it's going to provide specificity for what we are attacking. It's going to provide memory so being able to identify a pathogen that we've seen before, and self-tolerance, being able to distinguish self from non-self, which is very important so your immune system does not attack your own cells. We're going to talk a lot about antigens. Antigens trigger a specific immune system response. So antigens are anything foreign that gets into your body that your body says, whoa, this should not be here. That's an antigen. And there are two types of specific immunity. There's both humoral and cell-mediated. Humoral is going to focus on B cells producing those specific antibodies for that specific immune response. And cell-mediated is going to focus on T cells activating and targeting pathogens that are within cells or cancer cells. So humoral immunity is really focusing on extracellular things like a bacterial cell. Cell-mediated is focusing on T cells and focusing on cells that have been pathogens have gotten inside of the cell, or they're infected cancer cells and that cell has been altered in some way. So before we start talking about the different types of cells that are important for your adaptive immune response, let's just take a look at this diagram of the organs of your immune system. And I'm not going to have this on the test for you to label this at all. I just want you to see how important this is for your body and how many different areas are localized for your immune system, because this is going to include both your circulatory system and your lymphatic system for these different cells constantly surveying your body to find different pathogens that may have entered. And these are areas where those cells are concentrated. So places like lymph nodes and your spleen and your appendix and Peyer's patches are very important concentrated locations where these white blood cells can be looking for potential pathogens that have gotten into your body. And we're going to see here that bone marrow plays an important role for all types of blood-formed elements, including red blood cells, platelets, and white blood cells. So your bone marrow is where all of your blood-formed elements come from. You have hematopoietic stem cells in your bone marrow that can then mature and differentiate and develop into all the different blood-formed elements that you have. So that's going to include all those different types of white blood cells that we talked about. Remember that chart? So it's going to include your mast cells, eosinophils, macrophages, your B cells and T cells, and all of that. And it's also, though, going to include your erythrocytes and platelets. So all of the formed elements in your blood come from hematopoietic stem cells that are in your bone marrow. And just so you keep in mind where we're headed here, we're going to be talking about lymphocytes now, because these are part of the adaptive immune response, or the specific immune response. We're going to be talking about T cells and B cells. And these both, though, come from pluripotent stem cells or hematopoietic stem cells in your bone marrow. So they're all coming from those stem cells, but they're differentiating. Lymphocytes are exciting because now we get to talk about antibody production. And we'll see that a very specific lymphocyte is responsible for antibody production. So B cells are a type of lymphocyte that matures in the bone marrow. B cells, bone marrow. And then T cells mature in the thymus. T cells, thymus. Both of these, once they're mature, they're going to circulate through the blood and the lymph. So your circulatory system, where your blood travels to deliver oxygen and get rid of carbon dioxide, is very closely related to your lymphatic system. Your lymphatic system is really important for your immune system as well, and these B cells and T cells and other white blood cells will actually concentrate and congregate around lymph nodes in your body. And as your fluids are circulating through your system, they're constantly surveying for potential pathogens. And to be more specific, how is it identifying these pathogens or these foreign things in your body? By looking for antigens. Antigens are molecules that trigger a specific immune response. They're anything that your body sees as foreign. So an antigen could be a component of a bacterial cell wall, like peptidoglycan. It could be part of a bacterial capsule. It could be pili, flagella, proteins that are parts of viruses, fungi, or protozoans. So any part of an organism that looks foreign is the antigen, and then your body targets that entire organism. Food and dust can also contain antigenic particles. Most often they're going to be associated or connected with another particle that your body sees as foreign. And you can see at the bottom here, this artistic rendering of a bacterial cell. So the purple oval is your bacterial cell. And you can see on the surface, maybe this is parts of its capsule. This is what your body is identifying as foreign, not the bacterial cell as a whole, but some part of the bacterial cell is alerting your immune system that it should not be there. And so your body targets that entire bacterial cell by targeting the surface antigens. And you can see that it's targeting it using something called antibodies. Antibodies are specifically binding the foreign antigen. And we'll see more about antibodies in a few slides. So again, an antigen is anything your body sees as foreign. So it's not just bacteria, it's not just viruses, although they both can be antigenic. But it can also be your own cells that your body sees as foreign, like a nerve cell or a leukocyte or white blood cell. Maybe it's a cancerous cell, maybe something in it has been altered. Either way, your body does not see it as your self anymore. It's a marker of non-self when an antigen or a foreign molecule is present. How is your body going to determine if something's foreign or not? It determines the immunogenicity, or how immunogenic a molecule is, how likely it is to cause an immune response, by looking at its foreignness. So is this molecule an abnormal part of the body? Is it a normal cell or a normal component of the body? Its size, larger molecules are more likely to be identified by your immune system as foreign. That's why dust by itself is often not immunogenic. It's usually just not big enough to be identified by the immune system, but it can be associated with another component that is immunogenic, making it larger and more identifiable by your immune system. Its 3D structure. Structures that are more complex are also more likely to be identified by your immune system as foreign. And then finally, its composition. So the presence of certain proteins that should not be in your body, certain components that shouldn't be in your body. An example of this would be like lipid A in the outer membrane of gram-negative cells, or peptidoglycan. Our cells do not have peptidoglycan, so when our body sees it, we know that it is foreign. So by this point, I hope you're wondering, how is our immune system not attacking our own cells? We've talked about how there are foreign molecules, and it can identify foreign molecules, great, but that must mean that it can identify itself as well. And that's through something called the Class 1 MHC self-markers. And MHC stands for Major Histocompatibility Complex. And all of your somatic cells have a special collection of MHC markers that identify yourself compared to foreign cells. And your immune system knows this. And so it's able to coexist peacefully with your somatic cells by identifying those MHC markers. And this is known as self-tolerance. And for the next lecture, we'll see that this is not always the case though. Autoimmune disorders, when you have an immune system that is attacking your own cells, that's whenever your immune system stops recognizing your MHC self-markers on the surface of those cells. There are two types of MHC or major histocompatibility complex markers. There's MHC class 1 and MHC class 2. MHC class 1 is going to be on all of your body's nucleated cells. So it's not on red blood cells because they're not nucleated, but it's on all of your other cells and identifies your cells as self. They're your cells. This is also what happens whenever, after some type of tissue transplant has taken place and there's tissue rejection, it's from your body recognizing a different display of MHC class 1 markers on that tissue that's been transplanted. MHC class 2 markers, we'll see in a little bit, they're selectively expressed on special cells called antigen-presenting cells. And we're going to see these on both macrophages and B cells. So MHC class 2 is not on all cells. It's on special antigen-presenting cells. Just a bit of a background for antibodies, because antibodies are very important for the third line of defense and our specific immune response. Antibodies are also called immunoglobulins, or Ig. So these are all interchangeable. Antibodies, immunoglobulins, Ig. Antibodies are proteins that bind very specific antigens. So antibodies identify foreign things in our body, or antigens. Antibodies, we're going to see, are considered part of our humoral immune response and are related to B cells. And we'll jump further into the humoral immune response in a few slides. Antibodies have a lot of important jobs in our immune system. First off, there's opsonization. In opsonization, antibodies are coating some foreign object, so they're binding to lots of antigens on the surface and identifying it for a phagocytic cell, like a neutrophil or a macrophage, to come along and eat it. So opsonization is coating that foreign cell so it can be eaten by a phagocyte. Neutralization, certain antibodies can work as antitoxins and block the toxin from binding a cell, and we'll see an example of that on the next slide. Complement fixation, they can allow for complement binding. So remember all of those 25 to 30 complement proteins. So antibodies can help complement binding and help start the complement cascade. And then we have agglutination, and this is clumping together. So certain types of antibodies, especially IgM, which we're going to see in a little bit, can bind 10 antigens, clumps antigens together and alerts your immune system and brings antigens closer together so they're easier to get rid of. This image shows an example of both neutralization and opsonization. So first off, antibodies are binding specifically on this virus. It sees it as foreign, and that's preventing the virus from binding the host cell and infecting it. It's neutralizing the virus. Also, by coating the surface of this virus, it's alerting the immune system to come consume this virus and dispose of it. And so as it's coated with antibodies, that tells this phagocytic cell, this macrophage, to come along and phagocytize this foreign object. You have many types of antibodies that are surveying your system right now for an infection that you've come into contact with in the past. And you are able to have so many types of antibodies because they have two components. So there's this constant region, but up here is this variable region. It's this variable region that specifically binds to certain antigens that your body has seen in the past and has created certain antibodies for. And each antibody has this Y shape, and each of these arms at the top can bind one antigen. So an antigen can bind here and an antigen can bind here. So you have 5 distinct types of immunoglobulins, or antibodies, and some of them have different shapes. IgG, IgD, and IgE, and remember Ig stands for immunoglobulins, all have this singular antibody shape. IgA is a doublet, so it's 2 antibodies connected together, and IgM is 1, 2, 3, 4, 5 antibodies attached together. So that means IgM can bind 10 antigens at once, IgA can bind 4, and IgG, IgD, and IgE can each bind 2. Each type of antibody or immunoglobulin also has a specific job. IgA is going to be present in your secretions and fluids like mucus, tears, and saliva. IgD is going to be on B cells. It's going to be a B cell receptor that's actually in the membranes of B cells, and this helps stimulate the release of IgM. IgE is associated with mast cells and basophils and involved in allergic reactions. IgG is your most abundant antibody. It can make up to 60% of the antibodies that are going through your body right now and surveying your systems. And this one can also cross the placenta. And then IgM, you can see this is the pentamer. It's made up of 1, 2, 3, 4, 5 antibodies. This one is important for agglutination, so clumping of those antigens together. And it's also really important for your primary immune response. So the first time that you see a certain foreign thing in your body, IgM is highest and helps with the initial agglutination, clearing of the immune system and that foreign object, and fixing the complement proteins, those 25 to 30 proteins that are complementing the immune system and causing that cascade. Now we're going to talk about these 2 branches of the specific immune system, humoral immunity and cell-mediated immunity. So these are both parts of the specific immune response. And we'll talk about B cells, T cells, and then these other cells that are involved in the immune response. I want you to see this slide right here, right now, because you can see this TH cell or helper T cell is hanging out right in the middle here between the 2. That's because helper T cells are really important for specific immune response for both humoral and cell-mediated immunity. They activate both B cells and T cells, cytotoxic T cells. It's really important for activating both. And in terms of HIV, HIV specifically infects helper T cells. So you can see here why an HIV infection really is detrimental to your immune system, because it's targeting the cell that is important for activating both branches of your adaptive immune response. Now let's talk about humoral immunity. And for humoral immunity, we're focusing on B cells and B cells producing specific antibodies that target specific antigens. So B cells produce antibodies. Now let's see how a B cell is going to get around to producing antibodies. Over on the left here, we have an antigen-presenting bacteria. So that means there's bacteria in your system. It has antigens on the surface that your body sees as foreign. A B cell is going to come along and identify something foreign on that bacterial cell, and it's going to say, whoa, this guy should not be here. It's then going to use its MHC class 2 marker to display on the surface and tell your immune system, hey, there's something foreign here, and it's going to tell this helper T cell. That helper T cell is then going to release special cytokines, or that's the way that cells communicate with each other, right? Called lymphokines. And those special communication molecules are going to tell the B cell, oh yeah, you're right, that guy should not be here. You need to differentiate into an antibody producing cell. So the B cell differentiates into a plasma cell. And a plasma cell is an antibody factory. That's the type of B cell that's going to produce a bunch of antibodies. Now all of these antibodies can go off throughout your system and identify more of this bacteria that's present and mark it for your immune system. Another way that B cells can be activated and differentiate into a plasma cell to make antibodies is indirectly, by getting the information from an antigen-presenting cell. So it doesn't have to come in contact with the bacteria or the foreign object itself. An antigen-presenting cell can tell this B cell, hey, there's something foreign here and this is what it looks like. That B cell can then process that antigen and present it on its surface of its MHC class 2 receptor, and then show it to the helper T cell. The helper T cell will then release those cytokines or lymphokines in order to tell the B cell, yes, go ahead and differentiate. The B cell will then activate, differentiate into this plasma cell, and the plasma cell, again, is an antibody factory. It will make a bunch of antibodies specific to this antigen. Now we've seen that B lymphocytes, or B cells, because B cells are lymphocytes, a type of white blood cell, we've seen that they can be activated by these helper T cells to turn into plasma cells to make antibodies. But activated B cells can produce or differentiate into two different types of cells. A plasma cell to make antibodies to identify specific pathogens or antigens, but also memory cells. Memory cells allow our immune system to produce a faster reaction whenever we see an infection in the future and allow for quicker antibody production. Your body does make more plasma cells than memory cells, though, when these B cells are differentiating. Now, since we've talked about the different types of antibodies or immunoglobulins and how they're produced, let's talk a little bit about the primary and secondary immune response. And this is talking about when you are initially exposed to some type of antigen, that's your primary immune response. The next times that you see or are presented with that foreign antigen, that is your secondary immune response. During your primary response, the first time you've seen an antigen, the first time you've been exposed to it, it may take several days to mount a specific immune response toward it. But the major antibody produced will be IgM. That's that pentamer that consists of 5 antibodies stuck together. And then IgG will be the highest antibody. During your secondary response, though, any subsequent exposure to that antigen, IgG is produced more rapidly in larger amounts than IgM. So know that during your primary immune response, there are high levels of IgM. During the secondary immune response, there are high levels of IgG. So why do we care about that and differentiating between the primary and secondary immune responses? Well, let's say you have a patient and you're observing their antibody titer, or the amount of these different types of antibodies in their system. What would that tell you about the state of their infection? If you observe a high level of IgM, that will tell you that that is their initial exposure to that antigen, which is very useful in determining how that certain infection should be treated. Now on to the second branch of the adaptive immune response, cell-mediated immunity. And for this, we're going to be focusing on T cells, like cytotoxic T cells, and how they're activated and differentiate into those different types of T cells, each with specific functions. Now, what are these different types of T lymphocytes or T cells? Altogether, they're considered cellular assassins because they do kill cells. They kill bad cells that are considered foreign, and they're part of our cell-mediated immunity. Like B cells, they circulate in the lymph and the blood and migrate in lymph nodes where they can survey the fluids that are passing through to see if there's anything that shouldn't be there. Now, they are, again, part of our cell-mediated immune response because they act directly against antigens. So they're going to focus on intracellular pathogens or abnormal body cells like cancer cells, and they're going to kill those cells. Whereas the humoral immune response focused on extracellular organisms, like a bacterial cell. These are the different types of T cells we're going to talk about. We've already mentioned helper T cells and how they play an important role in activating both humoral and cell-mediated immunity. And there's also cytotoxic T cells, suppressor T cells, and memory T cells. All right, first off, we have our friends, the helper T cells, or CD4 cells. So called because they have CD4 receptors on the surface of the cell. This is actually the receptor that HIV binds to in order to infect these cells. As I've mentioned before, helper T cells play a central role in the immune response. They're really important for activating both humoral and cell-mediated immunity. Helper T cells are going to recognize antigens on APCs, or antigen-presenting cells, and then those antigen-presenting cells are going to activate B cells, macrophages, and also what we're interested in right now, cytotoxic T cells. Now, two types of lymphocytes are killer cells, both cytotoxic T cells and natural killer cells. We've already talked about natural killers in terms of innate immunity, but both cytotoxic T cells and natural killer cells work the same way. They both have granules inside the cells that contain chemicals called perforins. When they make contact with their target cell that they want to destroy, they release those chemicals and lyse the cell, killing it. So both of these work the same way, but they're activated differently. Natural killer cells are part of our innate immune response, and they're looking at the MHC class I receptors on cells to identify self from non-self. Cytotoxic T cells, on the other hand, have to be activated by a helper T cell. Thus, they're part of that third line of defense and our adaptive immune response. Now, how are cytotoxic T cells activated? Let's go through these steps. First off, we have antigens. We have something foreign in the body. That's going to be phagocytized or consumed by a macrophage. The macrophage is then going to process that antigen and present it on its surface using an MHC class II receptor. And that MHC class II receptor is going to be recognized by a resting helper T cell. Helper T cells can only recognize antigens using this MHC class II receptor. The helper T cell is going to recognize, yes, there is something foreign in the body. Let me activate. So the helper T cell activates, and then it's going to activate other macrophages to go consume other bad things in your body. At that point, cytotoxic T cells can become activated by the presence of MHC class I receptors that are presenting that antigen. Cytotoxic T cells can only be activated by MHC class I receptors. This activated cytotoxic T cell then knows what it's looking for, and it can go around and bind MHC class I receptors that are presenting that same antigen on the surface of infected cells. Once it's made that connection, it will release those chemicals inside of it and then burst and lyse the cell, killing it. Sometimes our normal cells can turn into cancer cells, maybe because the cell is exposed to a mutagen or it made some mistake replicating. Some of the antigens on the surface of this cell changes. These cells, like a lot of body cells, are constantly shedding off bits of protein from their surface into the circulatory system. And oftentimes tumor antigens are among these shed proteins, especially if it's on a cancer cell. So these shed antigens prompt action from the immune defenders that you have, like cytotoxic T cells and natural killer cells and macrophages. And according to one theory, these cells are just constantly patrolling the immune system and provide a constant body-wide surveillance, catching and eliminating these cells that have undergone malignant transformations. Tumors do end up developing, though, when the immune surveillance breaks down and is overwhelmed and does not catch early signs of cancer cells. Now, the final type of T cell that we're interested in is regulatory T cells. And these T cells are very important because we need a way to regulate the immune response. We need a braking mechanism or a checkpoint to prevent the immune system from overexpressing in response to some type of antigen. This is how autoimmune diseases could flourish if there's no way to stop that immune response. We don't fully understand yet how regulatory T cells operate, but these are the two major theories. One, T cells could compete for the same antigens, so they could block off these receptors and stop white blood cells from being activated. Two, T cells could compete with cytokine signals. So as T cells are proliferating and you get more and more T cells, they're releasing more and more cytokines, and those cytokines are giving chemical signals to those white blood cells to stop the immune response.